Before I get started, let's go ahead and make sure we're recording, screen is sharing, everything looks good. Alrighty. So in chapter 3, we're talking about microscopy. Okay, so we're going to be talking about observing microorganisms through a microscope. So the first organism here is Helicobacter pylori. So when looking at many of the images in our textbook, we're going to be looking at these things called micrographs. All right. So this is a micrograph using a fancy microscope that we're going to talk about in just a moment to allow us to visualize the structures of this particular microbe. So Helicobacter pylori is the causative agent for stomach ulcers. So Figure 3-2 in our textbook talks about the microscopy ranges that we see. So it's important for us to understand that there's a range in which we can see things with the unaided eye. All right. So that means without any type of tools such as a microscope or a handheld little lens. So with the unaided eye, we can see for some people about 1 meter to about 100 micrometers in size. Just depends upon your vision, right? So this just shows us a tick. It's actually the actual size of a tick. And we know that many times you don't know a tick is on you until it's on you, until you feel it, right? So that's pretty small. So comparing that to red blood cells, which we can use a light microscope to look at, how do we know that we can use a light microscope? Well, we see right here that it has the letter LM, that stands for light microscope, and it's showing us the scale at which these organisms are pictured. So this is 6 micrometers. If you just take that little scale there, it'll be 6 micrometers. And so we can use a light microscope from the range of 200 nanometers until about 10 millimeters. All right. And so then we have this overlap for these electron microscopes. So we have scanning electron microscopes and transmission electron microscopes. So for scanning, you'll see SEM, scanning electron microscopes, and then TEM, transmission electron microscopes. And then the atomic force microscope, AFMs, are going to be able to look at specimens at a much smaller capacity, right? So these are very small. E. coli bacteria, you can see them on light microscopes as well, but the scanning electron microscope gives you a really beautiful three-dimensional view of them, whereas your transmission electron microscope allows you to see the internal structures really nicely. And then this atomic force microscope here allows you to see really small things that you would not be able to see even with your electron microscope. So your DNA double helical structure as seen here. Now, in our course, many of our organisms will fall within this range right here. So about 10 micrometers until about 10 nanometers, that's going to be the size of these organisms. So one of the first things we're going to be doing is talking about units of measurement, right? So this should be a review for you guys, right? So we're going to list the units that we use to measure microorganisms. So we know about the metric system. And so the metric system is what's primarily used across the world. I've told you guys before that here in the US and 2 other countries, they use the imperial system. And so for the metric system, we know that we have kilos, we have the hectares, we have the deca base, the deca measurement, we have the base measurement, we have the deci, we have the centi, and we have milli. All right. And so we're going to be going from large to small in these quantities. And so when looking at this, many of our microorganisms aren't even pictured right here, right? So this particular number, when we look at these values, kilometers, when we look at it, it reference point to the base measurement. So the base measurement is 10 to the first power. 10 to the 1. So that gives you the 10 to the 1 whenever you raise something to that. Well, I'm sorry, 10 to the 0. I'm sorry. So whenever you raise something to the 10 to the 0, that's going to be a 1. It's a null measurement. So 10 to the null will give you 1. And so 10 to the 1 will give you the decameter. If we're looking at meters, the hecta will be 10 to the 2. And then the kilos will be 10 to the 3. And then you have a deci. So deci is we're going to be still 10 to the negative 1 because you're going to be increasing in its size. Centi will be 10 to the negative 2 because relative to your meter, it's going to get smaller as well. And then your milli is going to be 10 to the negative 3. All right. And so these values are all lowercase. So kilos, all these letters are lowercase. I just wrote them in capital letters just for the sake of writing it out on this slide. And then you have micros. So micros are really small. Micros are 10 to the minus 6. And then your nanometers, nanos are going to be 10 to the negative 9th power. All right, so it's showing you relativity here. So 1 micrometer is equivalent to 10 to the minus 6 meters, which is equal to 10 to the minus 3 millimeters. And that's because to go from 10 to the minus 3 to 10 to the minus 6, you're going to be just increasing that size by 3. All right, to go from decrease, should I say, because you're getting smaller. With nanometers, 1 nanometer is equal to 10 to the minus 9 meters, which is also equivalent to 10 to the minus 6 millimeters. All right, same thing. Going from this 10 to the minus 9 meters to the 10 to the minus 6 millimeters, you're going to be getting smaller in that particular value. All right, so 1,000 nanometers is equivalent to 1 micrometer, and then 0.001 micrometers is equivalent to 1 nanometer. So in our course, we're not doing the calculations, okay? So we don't do the calculations here. We do that in the lab. However, it is important to understand that. So I would like you to still practice that so you can understand what this looks like. All right, so practice question. How many nanometers is 10 millimeters? So you pause the video to answer this question. We are going on. All right, so the correct answer for this question is going to be B as in boy. All right, B as in boy, 10 to the 7th. So when looking at the microscopy, for this particular example, you have your things we're going to talk about with the diagram that's going to show the path of light through a compound microscope. We're going to look at total magnification as well as resolution. All right, so first things first, when talking about microscopes, remember, I said they started out very simple, right? Very, very simple. The simple microscope that we saw with Antoine van Leeuwenhoek, it had one lens, right? So it only had one lens as seen right here. So you see that you could put the specimen on the tip of the pen. And once that specimen is there, you're able to look at it through the lens. You can position it differently by using the screws to move it, adjust it, and all that good stuff. But for the most part, you're able to visualize this particular specimen at a higher magnification compared to a magnifying glass. So when talking about light microscopes, you have different kinds. These light microscopes, what they have in common is that they use visible light to observe specimens. All right, that's what they have in common. So these include our compound light microscope, which is what we have in our lab, your dark field microscope, your phase contrast microscope, differential interference contrast microscopes, fluorescence microscopes, and confocal microscopes. Now in our course, I don't make you know all of them. On your study guide, I do narrow it down for you guys to focus on specific microscopes. All right, so looking at the structures of a microscope, we went over this in lab, so I won't hit too many details of it because you guys did this in lab. I checked your grades for it already in lab, and it's just a little just a little review real quick. So ocular lens is what we're going to be looking through. That's going to magnify the image for us as we look at that particular specimen. So the ocular lens is going to be held in the body tube right here, connecting there, but also within the body tube, there's a prism that's going to hang out within that the area, and that's going to be able to allow the light to refract and bend to be able to be captured from the objective up through the ocular. So here we have the revolving nose piece that holds the objective lenses. You have a stage at which you're going to be putting your slide on the stage, and the stage clip secures that on the slide. The arm is where you're going to hold this to basically be able to move the microscopes. We hold the arm and we also support it on the base so we don't drop the microscopes. You also have to turn your microscope on, right? So you have to turn your light microscope on to get the process going. And so the light microscope switch should be somewhere on it. This is a light intensity knob right here. It controls the amount of light that actually reaches the specimen. And so here you have the condenser. So the condenser lies underneath the stage, and that's going to help focus the light through the specimen. The iris diaphragm, that lever is going to control the amount of light that actually enters the condenser itself. And so then we have the illuminator, which is the light source. And so in order to look at the slides, you have to have some knobs, right? So you see this knob over here on this side. This knob right there is going to be your mechanical stage knob. It allows your stage to be moved front to back and then right to left. Whereas your coarse focusing knob allows your slide to be able to be moved up or down, right? And we talked about the coarse focusing knob. We use it only on certain objective lenses. And then your fine focusing knob is going to fine tune your specimen. All right, so in your compound light microscope, we talked about we have to calculate total magnification, right? So for total magnification, you are going to have to look at the objective lens magnification, and it's going to be multiplied by the ocular lens magnification to give you the total, right? So notice that our microscopes in our lab have ocular lenses that measure at 10x, but we have different objective lenses. So we have 4x objective lens, we have our 10x objective lens, we have our 40x objective lens, and then we have our 100x objective lens. So to calculate the total magnification, you simply multiply each value by 10, right? So the total magnification for the 4x would be 40, right? The total magnification for your low power lens would be 100, right? The total magnification for your high dry lens right here would then be 400, right? Because this is a review. We talked about this in class. And then your last one, your oil immersion lens, would have a magnification of 1,000. All right, 1,000. All right, so when talking about the direction at which your light's going to travel, remember you have a light source. So you turn the light on and the light's going to come from the illuminator, which is going to pass through your condenser lenses. That will then reach your specimen, reach your objective lens, that will ultimately reach in the body tube, hit the prism that's present, and the prism is going to redirect the light flow throughout out towards the ocular lens for us to visualize that specimen. All right, so when looking at this ability to look at these different points, we have to understand the term resolution. So resolution is the ability to distinguish between 2 points, right? So basically allowing us to see the fine details and structures of a specimen. So when talking about a microscope that has a resolving power of 0.4 nanometers, it has the ability to distinguish between 2 points that are 0.4 nanometers apart, right? Like looking at that, those 2 points right there, that's really, really, really small. And so a light microscope will be able to differentiate between how close those specimens are. All right. And so shorter wavelengths of light provide greater resolution. So remember, wavelength is going to be a measure of the light of the, well, when looking at the electromagnetic spectrum, of course, the electromagnetic spectrum is going to exhibit a range of energy, and it's going to be organized by wavelength and frequency. And so we only focus on the visible light spectrum, right? So with visible light, that's going to be a certain very small number. Energy rays in that particular area is not going to be as energized, right? They're going to be shorter wavelengths, and they have the ability to provide greater resolution, though. Okay, so the limit of a resolution of a compound light microscope is 0.2 micrometers. So our compound light microscopes have the ability to differentiate between 2 points that are 0.2 micrometers apart, right? Now, the limits of magnification for a compound light microscope can be up to approximately 1,500 times. So that just depends upon the scope that you're looking at, right? Ours is, as we just calculated, 1,000, right? So we also have this thing called the refractive index. So with the refractive index, you have to it is referred to as the light bending abilities of a medium. All right. So the light bending ability of a medium, the measure of that particular value. So when talking about the light, it can refract after passing through a specimen to an extent that it does not pass through the objective lenses. All right. It does not pass through the objective lenses. And so when looking at the immersion oil is pretty important because immersion oil allows for the light to not refract. All right, so the immersion oil is really good at allowing the light or keeping the light from refracting. All right, and so let's talk about that. So when looking at this particular example, I studied the fact that we have a microscope, the objective lens here. And so you're going to be trying to look at this specimen on the 4x, you look at it, good. 10x, you look at it, good. And then you get to the 40x, it still looks good, right? But if you look at the specimen under the 100x, the oil immersion objective lens, without using oil, most of that light is going to refract and bend away and not travel through the objective lens. You're going to lose a good bit of that. So what happens is you add the immersion oil because when we look at refractive indices, refractive index for the following, well, the refractive index for these different mediums are as follows. So the refractive index, kind of get that written out, does not have any, it does not have any units. So refractive index, refractive indices do not have units. All right. It is just a number that's going to be a ratio. And so for air, its refractive index is 1.00. All right. But for glass, its refractive index, specifically when looking at the microscope slides, their refractive index is 1.517. So this is the case for standard common types of optical glasses and transparent materials, whereas oil, and specifically immersion oil that we use, so immersion oil has a refractive index of 1.518. All right, 1.518. Okay, so when looking at this, we see the light passing through, right? The slide goes to the slide, and when you do not have any oil present, it bends away. And so that's because you have the glass at 1.517, the air is at 1.0, and that's a big jump, right? That's a really big jump. But when you add the oil, it kind of smooths out that transition. So you simply have the light passing through the glass at 1.517, then 1.518, and the oil touches the objective lens. So that way it doesn't get lost to the surrounding environment. It just simply passes through and reaches that lens. All right, cool stuff, right? So let's jump into our microscope. So I told you guys before, we have compound light microscopes, and we have this thing called bright field illumination. So bright field illumination is typically what we use because it allows us to see dark objects against a very bright background. All right, so when this happens, the light is reflected off the specimen, and it does not enter the objective lens. So light reflected off the specimen does not enter the objective lens. So it may be difficult to look at unstained cells as a result because we can't really see the contrast, right? So the unstained cells typically are harder to look at. That's why we stain them. We add different reagents to look at them, to visualize them. Yeah. So here we see a paramecium here on the bottom right-hand side. And this paramecium is being looked at. We're going to look at this as like our model image across the different types of microscopes. So the light microscope for this particular paramecium, it shows the contrast. You can see this against that background, right? You can see the internal structures nicely. You can see the outline, the external membrane of this little fella. So the external membrane is present. It's called a pellicule. You also see like these little small structures emanating from the body. That's the cilia, the hair-like structures that aid their movement, right? That allow them to move. So we're also looking at the light path of our bright field microscope. So light source is going to allow the light to pass through the condenser lens, to the specimen, reaching the objective lens, ultimately hitting the ocular and allowing us to visualize that specimen. Yeah. So there's a nice little video. You guys can watch it on your own time. So now we're going to talk about the different types of microscopes, okay? Dark field, phase contrast. Just to name a couple, we're going to also compare and contrast our electron microscopes from our light microscopes. And then we're going to look at the uses for these electron microscopes, scanning and transmission, as well as the scan probe microscopes. All right, but first, the dark field. So if the bright field looks at specimens, the bright field looks at dark objects against a light background, the dark field has a dark background, right? Dark field. So here we're looking at light objects against a dark background. And so here they typically put an opaque disc in the condenser to allow us to see that dark background. It makes the background dark and you get to see the lighter objects. So only light reflected off the specimen here actually enter these objective lenses. And so we typically use this microscope for viewing live unstained microorganisms, right? Because you can see those unstained microorganisms against the dark background. So you can use it to look at Treponema pallidum, which is the causative agent for syphilis. So this spirochete, the spiral-shaped bacterium, can be visualized using a dark field microscope without even staining it. So here we see what the paramecium looks like, right? So if we look at the paramecium here, right, it's going to have that external covering, just like we saw. We can see like little small structures there. That's still the cilia emanating from the body. And then we have like the internal components, right? And then notice how the background is dark and the specimen itself is like really light, right? It's like almost glowing, right? But it's not. So here we see the path of the light. You have the light source. Some of that light's blocked because of the opaque disc, right? So some of the light is blocked, but the light that does pass through will reach the condenser lens, hit the specimen. Some light will be reflected off the specimen, and it's going to be captured into the objective lenses, while some will be unreflected and it goes away from the objective, so it's not captured. And then you're ultimately able to visualize the specimen. All right, so now we're going to switch gears to talk about the phase contrast microscope. So phase contrast microscopes allow us to see detailed examinations of living organisms and their internal cell structures. So for these guys, no staining or fixation is necessary. Okay, so we're going to talk about staining and fixation. Fixation basically means the specimen is adhered to the slide. So when we use bright field and bright field and dark field microscopes, many of the times we fix the specimens. In some cases, we stain them. All right, whereas for this one, you don't have to do any of that. So here's going to use two sets of light rays, direct rays and diffractive rays to form an image. So let's see. So here we see the light path where we see the light source having an annular diaphragm present. So this annular diaphragm is going to be present, and this little ring-like structure is going to have the light pass through. And so the direct light rays are going to travel a different path from the light rays that are reflected or diffracted. All right, they're going to be traveling differently. And so these rays, the undiffracted light that was unaltered by the specimen, is going to be recaptured to be combined for us to visualize at the eye. So reflected or diffracted light rays are indicated in the blue, and then our direct rays are indicated in the red. All right. So when looking at the specimen, we see a lot of good details, right? It looks really cool. So you can see some internal structures really nicely. You still see the pellicule, the external covering, the external membrane. You also see the cilia present on the surface, right? So this allows us to see a really nice detailed view of the specimen with a higher differentiation. All right. So we're still using light here, right? It's still using light here. Okay, so I want you guys to differentiate or answer this question. How are bright field, dark field, phase contrast, and fluorescent microscopes similar? So think about that. All right, think about that. Really, how are bright field, dark field, and phase contrast microscopy are similar? Yeah. Okay, now we're going to talk about electron microscopes. So electron microscopes use electrons, right? Instead of light, which makes sense, right? So the shorter wavelength of electrons gives the greater resolution for these specimens. Now, we typically use electron microscopes to see stuff that's too small to see with the light microscope. So viruses are too small to see with light microscopes, with the exception of like Ebola. Ebola is pretty big, so you can probably use a light microscope for Ebola, but that's not the case for everything, right? So that's why many labs or institutions have electron microscopes. And so you can also see internal cellular structures using an electron microscope, detailed structures. And so instead of, again, I said they use electrons, they're going to be using electromagnetic lenses to focus the electron beams. So one thing about electron microscopes, the graphics are going to be called the micrographs, the electron micrographs. Now, many of these images are black and white, all right? Many times we add the colors to it to make it more aesthetically appealing for what we're talking about. It's very rare that you'll see color already there, like with certain bacteria, but it does happen. So Staphylococcus aureus is naturally gold. It's naturally going to have that pigment, so you don't necessarily have to enhance that, but it is to kind of give a more aesthetically appealing look for the surrounding environment too. So here we have the transmission electron microscope, our first one. So this particular scope uses a beam of electrons that are passed through ultra-thin sections of a specimen, then through an electromagnetic lens, and then focused by a projector. And so when looking at these guys, you typically look at the specimen through the projector. The images are produced on a viewing screen and we save them digitally. All right. Now, they also stain these specimens with heavy metal salts. So I wonder, think about it. If we're staining them with heavy metal salts, are these specimens alive? You're like, no. Well, we knew that too because we're looking at the electron microscopes, which use electron beams. So we don't really pass electron beams through living tissue. So that tells me right there, like, yeah, most likely to be a prepared specimen. So they stain it with heavy metal salts for us to see it. So this one, the transmission electron microscope, has a very high magnification of 10,000 to 10 million times. That's amazing. All right. Its limit of resolution is 0.2 nanometers. So that means that it has the ability to see 0.2 nanometers apart when looking at a given specimen. All right, so the transmission electron microscopes I told you guys before, these are very, very expensive. They can cost up to, I told you that we got a really expensive one on our main campus, but the price can range from like a few $100,000 to a couple $1 million. Yeah, very, very expensive. You can buy them refurbished too, and that'll be less expensive, but still very costly. So how do you prepare the specimens? Well, you can do that through fixation, like I told you guys before, where you adhere the specimens to a slide, or you will dehydrate them so you can get tissues, dry them out, and slice them into very thin sections. So histologists, or histology techs, they do this type of work, prepare specimens to be visualized by another personnel member. And so these specimens are viewed under a high vacuum environment to basically minimize contamination, but also to allow the specimen to be in the form that they're trying to look at it in. So that's why the preparation kills the specimen, and it may cause shrinkage and a little bit of distortion that could leave a little bit of artifacts in the specimen. And the thing too is that sometimes these artifacts can be markers of the people that do the work. So like there are some people that do the work so consistently that if you see their work, you're like, oh yeah, so and so did that because they have like certain things that they do. There's certain techniques, should I say. All right, so figure 3-12A shows us this paramecium steel. And you're like, whoa, that looks completely different. So this is a colorized image that shows us paramecium. And so we can see the nicely like laid out internal structures, right? So this is the some type of vacuole, so contractile vacuole, these different organelles on the inside because paramecium is a eukaryotic organism. So we see like, oh, the pellicle is there, right? You see like the small cilia is really faint, but you can see it. But yeah, so really nice, very nice. So looking at the pathway of the electron beams, you have an electron gun that's going to allow the electron beams to be released, and then it's going to pass through or pass by the electromagnetic condenser lenses, which will then reach the specimen. After passing the beams to the specimen, the objective lenses will then allow it to be picked up to by the projector, and you'll be able to view the specimen through the eyepiece, right? And then capture an image. Capture an image. Now let's look at the scanning electron microscope. So these guys are a little bit different. This is where you're going to be using an electron gun to produce a beam of electrons that scan the entire surface of the specimen. So scanning electron microscopes, they scan the surface to give us or render us a three-dimensional view of the specimen. All right. So how do they do that? Well, they use secondary electrons emitted from the specimen. They're going to be transmitted to an electron collector, then amplified to be used to produce an image for us to look at. So this particular scope has magnification of 1,000 to 500,000 times. All right. So smaller, right? Smaller than our transmission electron microscope, which makes sense. And so this one has a limit resolution of 0.5 nanometers, right? So a little bit bigger than our transmission electron microscope, but still much smaller than our light microscope. So with the scanning electron microscope, these aren't as expensive, but still expensive, right? These guys can be about $50,000, and some can go up to $1 million. Yeah, so those are less expensive than the transmission, but still costly, still very costly. All right, so looking at our scanning electron microscope, figure 3-12B as in boy, we see our paramecium, right, on the right-hand side, and see how lovely that is. We see the outside external view. We have like this little gap in the structure, the cytostome. You see like the little cilia around the surface of it. So the pellicule, you can see like this three-dimensional view of it on the outside surrounding the cell. And so this gives us a really nice overview of that structure compared to so far we've been seeing the two-dimensional view, right? So far we've been seeing two-dimensional views. This is the first three-dimensional view courtesy of our SEM, scanning electron microscope. And so here we see the electron gun is going to pass out those primary electron beams that pass the electromagnetic lenses that will ultimately allow us to use the viewing screen to visualize this. And then we're going to have the electrons being collected from that specimen, right? So the electrons are going to be captured from that specimen, collected, amplified, and then transmitted onto a viewing screen for us to look at, right? So again, that's when we can start taking pictures of your specimen, right? So there's a lovely little video. You guys can watch that. All right, so question, why do electron microscopes have greater resolution than light microscopes? Think about that. All right, think about that. All right, now to the fun stuff. Okay, so now we're going to talk about how we prepare specimens for light microscopy because you will be doing this in the lab. Okay, so I want you guys to be able to differentiate between an acidic dye and a basic dye. We want to talk about the purpose of simple staining. Then we're going to talk about differential staining. So specifically the gram stain steps, the protocol, what the cells look like after each step. We're going to talk about the acid fast stain as well, and the capsule stain, endospore stain, and flagella stain. Okay, but first things first, what is staining? So staining is when we color microorganisms with a dye, and this allows us to visualize certain structures of that microbe. Okay, but first things first, you have to prepare a smear, right? So the first thing you have to do is prepare a smear to of the microorganisms on the side to look at it, right? So to do that, we're going to ultimately have three steps, right? So, well, a few steps. So the first thing with making a smear, just want to write this down. So to make a smear, a couple things. You want to get your microscope slide. You want to wipe it off with a alcohol wipe to clean it, right? We keep alcohol wipes at our stations in the lab. And so once you wipe the slide down, we're going to consider it clean. And then we're going to take our inoculating loop. And so our inoculating loop, we're going to see in lab, it's going to be a metal structure. It's going to be metal. And it's going to have like a little loop on the tail end of it, like a real loop. And so with the inoculating loop, we're going to add a drop of water to the inoculating loop. And so once you add a drop of water to the inoculating loop, you're going to add a drop of water to the slide. All right. So you add a drop of water to your slide. All right. So you add a drop of water to your slide, and then you're going to then sterilize your inoculating loop again. And once you sterilize your loop, you're going to then capture a little bit of your bacteria. Right. So you want to sterilize your loop. Trying to get this for you guys, sterilize loop. And then once you sterilize the loop, then you're going to, as I mentioned, get some bacteria on the loop. And then once you have the bacteria on the loop, you add bacteria to that drop. So you add bacteria to the water on the slide. All right. You add the bacteria to the water on the slide, and then you mix and air dry. All right. So once you add the bacteria, you want to then mix it with the water. And then once you mix it, you're going to then allow it to be air dried. And once you air dry it, it's going to be ready for fixing to the slide. All right. So you're making your smear by following the steps. I didn't write all the steps down, right? I was saying it. So I would go ahead and write down the steps that I was saying. I couldn't write everything because I knew I didn't have a lot of space on the slide, but please make sure that you guys take all the steps. So the last thing would be you're going to heat fix it, right? So after it air dries, air drying takes a very long time. So this is why I'll give you some tips. So when you fix the slide, well, as you're preparing the slide, I would say that you want to not put too much water, just a small drop of water and a small amount of bacteria. The more water you put, the more bacteria you put, the longer it's gonna take to dry. All right. So once you heat fix it, you always have to heat fix before you stain your specimen. And that's for a couple of reasons. The fixing allows the microbes to be attached to the slide. Fixing kills the microorganisms, and then fixing will preserve parts of the microbes with minimal distortion. So it allows you to see the structure that you want to look at. All right. So how can we fix? So chemical fixation is cool. We don't do that. That's a lot of times they do this with tissues. Like histologists use that methanol for a minute to to smear to make a smear with their tissues. But we use heat. All right. So for the heat, remember I was saying after you prepare the specimen, you want to pass the smear through the flame. And so you'll pass it through the Bunsen burner several times to gently heat it. Now you don't want to do it too long. You want to waft it through there. So that way you don't burn the specimen off. And I'll show you guys that in the lab too. So there's a lovely video for you guys to watch. Watch it when you have a moment. Now, let's talk about the last little bit of making these smears. So we have to understand what the stains consist of, all right? So stains consist of a positive and a negative ion. So what are ions? Ions are charged elements, right, on the periodic table. So ions basically when you have more positive or negative charges. It depends upon what we're talking about. So when talking about stains, these stains are referred to as chromophores. All right, so these stains are referred to as chromophores. And so in a basic dye, the chromophore, right, the charged ion is a cation. So what do we know about cations? Well, cations are positively charged, right? That T to me looks like a plus sign, so we're going to go with that. So cations are positively charged. And so when looking at this, these positively charged ions, right, will be present within these different stains. And so crystal violet is a basic dye, methylene blue is a basic dye, and saffronin are going to be basic dyes as well. So that means that these are going to be positively charged dyes, right? These basic dyes. Now, in the acidic dyes, these are going to be chromophores as well, but these chromophores are going to be anions. So anions are negatively charged. All right. So anions are negatively charged. And so with these anions, these include eosine, acid fusion, and nigrosin. Okay. So what do we know about bacterial cells? Well, bacterial cells have a overall negative charge. And the reason for that is because of the hydrophilic head region, right? So it has those hydrophilic heads, those hydrophobic tails, but the hydrophilic nature of this particular cell membrane allows it to readily interact with the external cell components and as well as the internal cell components. So that gives it that negative charge. And so one thing I've learned about charges is that opposites will attract, right? So those opposite charges, such as the negatively charged membrane, will allow for the cations to be connected to that. All right. I want to erase that. I want to do another color. All right. So the positively charged cations will be able to interact with our negatively charged cells, bacterial cells. So that's why bacterial dyes will adhere to the cells. All right. Basic dyes such as crystal violet, methylene blue, and safranin will adhere to the bacterial cells. Whereas your negative, right, dyes, your anions, the acidic dyes, these guys are going to repel, right? Because we know that like and like charges will repel, right? So like and like charges are definitely going to repel. And so with these charges being repelling, they're going to stain the background. So negative charges will stain the background, and that allows us to visualize whatever structure that they're not staining. So negative staining will stain the background instead of the cell itself. So we'll see that with the eosine, acid fusion, and nigrosin. All right, so let's look at some simple stains. So simple stains are going to basically be the use of a single basic dye. All right, the use of a single basic dye. And so when looking at these examples, that basically means I'll use one stain such as methylene blue. I could use carbofusion. I could also use crystal violet or safranin just to stain or highlight a certain feature. All right, so this highlights the entire microorganism to visualize cell shapes and structures. Now, we can also have a mordant. All right, a mordant is like it works in combination with a stain to kind of hold it in place. So when we look at our protocol for gram staining, we're going to see crystal violet and iodine forms a mordant, and that's allowing the cells to be held together and enclose the specimen to make it look bigger. So here's a video of the animation for staining. I did play it for you guys during class, so I would like you guys to look at it again. So remember, you can click on this one. This is a clickable link. Let me turn my pen off. Or matter of fact, let me come out the presentation. All right, so if I come out the presentation, you can click on the video, click on it, open hyperlink for you guys, and you'll be able to watch the video. So let me see right quick. Yeah, so you guys should be able to watch the video when you click out of it. All right, good deal. Okay, so we have differential stains. All right, we have differential stains. So differential stains basically means we're trying to differentiate or distinguish between bacteria. All right, we're trying to differentiate or distinguish between bacteria. So what stain protocols are doing this? We have gram stains and acid-fast stains. All right, gram stains and acid-fast stains. And so when looking at these protocols, let's look at the very first one, gram stains. So gram stains allow us to classify bacteria into one of two categories. It's going to be either gram-positive bacteria or gram-negative bacteria. Okay, so gram-positive, these bacteria will have a thick layer of peptidoglycan in their cell walls. So like we have our cell membrane, plasma membrane, and our cell wall, and we have a very, very thick layer of ours in our cell wall of the peptidoglycan. That's not a good example. I don't like that one. I'm going to erase that one. I'm going to wait till we show the actual pictures because I don't like that one. So let me erase it because I don't like it. So we'll look at that one in more details when we have a better image. So with that one, thick peptidoglycans cell walls and they're going to stain purple. Okay, so gram-positive bacteria are going to be purple. And so when looking at our gram-negative bacteria, these guys have a very thin layer of peptidoglycan in their cell walls. Now, I can draw the structure for them in that they have their regular old plasma membrane, they have their regular peptidoglycan layer, and then they have their outer membrane. So our gram-positive cells will just have its little membrane and its little cell wall, whereas your gram-negatives will have like an additional layer, that outer membrane of LPS, so lipopolysaccharides. I will be saying it. I won't make you guys say it, but I will reference it as LPS, lipopolysaccharides, and we'll talk about what that word means in a little bit. And it also has the phospholipids, right? Because we know the phospholipids represent the cell membrane. So these guys are going to stain pinkish red. Sorry. So these guys, the gram negatives, are going to stain pinkish red. Okay. Gram positive for purple, gram negative for pinkish red. So let me tell you how I used to remember it in school. I was like gram positive for purple, P for purple, and then the negative, the red right there, that N and the R, they look kind of close. If you continue that N on, it makes the R on, it looks like an N, right? If that's one way you want to remember it, or you can just remember it like, regularly. Okay, so let's go through the steps. So when you go through the steps, you'll see that we have some reagents that we're going to be using over here. Okay, so let's talk about this. So in any type of differential staining protocol, we're going to have a primary stain. So the primary stain here is like the first stain. So the primary stain is going to be crystal violet. So in a gram stain protocol, the primary stain is crystal violet. All right, so the primary stain is crystal violet. And so crystal violet is going to be, I should probably use a better color. Crystal violet is actually going to be violet or purple, should I say. So crystal violet will be violet or purple, right? And then once you add the crystal violet, it's going to be for 60 seconds. So you add the crystal violet to your slide for 60 seconds. And then once you are finished with the crystal violet on your slide, then you're going to rinse it with water, right? So you're going to rinse it with water, and you're going to rinse it with water until it runs clear. All right. And so then you're going to add your iodine. All right. So then you're going to add your iodine. So iodine is actually going to be brown in its color and you're going to apply it for 60 seconds. And so the iodine, the Grams iodine acts as a mordant. It's going to form a complex with the crystal violet in the cell's peptidoglycan layer. It's going to form a complex. All right. And so the color is still purple, right? Now, when we finish with this particular 60 seconds, then we're going to rinse the slide with what? Water. So we rinse it with water until it runs clear. Okay, the very next step is really important, guys. So the next step is kind of critical. So the next step is, I know this is yellow. I'm going to probably write this in another color, but I wanted to start off within the yellow so you guys can see, like I'm trying to highlight that. So I'll change the color to this, let's say I'll do a green right quick just for the interest of us writing. So the decolorizing agent is going to be most likely alcohol for gram stain. So we typically use alcohol, specifically ethanol. And I would say for this one, 5 to 7 seconds. All right. Very, very fast. I would say 5 seconds because what ends up happening when you do this protocol, by you guys being kind of new to it, it's going to take you a few seconds to pick up the alcohol, get it ready, pour it, and decolorize that specimen. We'll get the water cleared off from after you finish decolorizing. So I would say about 5 seconds. And then once those 5 seconds are up, I will go ahead and rinse it with water until it runs clear. All right. Once you hit 1, 2, 3, 4, 5, rinse with water, 6, 7, right? So by the time you reach to that 7, you'll be already in the process of getting it off. You'll be probably in the process of getting the alcohol off at that point. All right, so the last step is where we're going to add safranin. Safranin is going to be the counter stain. So crystal violet is the primary stain. Saffron is the counter stain. So that means your primary stain is going to stain them one pigment, whereas your counter stain will be another color. So in this particular case, your saffron is going to be that pinkish red color that we talked about. And we do that for 60 seconds as well. 60 seconds. And then once you have finished those 60 seconds, what do you think you do? You're going to rinse it with water until it runs clear. Now, note that your gram-positive bacteria are going to be purple the whole way through, right? So your gram-positive bacteria are going to be purple after adding the crystal violet, purple after adding iodine, purple after adding alcohol, and purple after adding your safranin. Okay, spectacular, right? All right, so for our gram-negative bacteria, we see that we have the same reagents, right? So we see in this particular example, once we add the crystal violet, it's going to be purple, right? Or violet, right? So it's going to be purple or violet. And so then you have your grams iodine added to it. And so with the grams iodine added to it, you're going to then see the color still going to be purple, right? For gram negatives. All right. So it's going to be purple for the gram negative. And then you're going to add your decolorizing agent, right? So far, 60 seconds for the crystal violet, 60 seconds for the iodine. Rinse between each step. Rinse after the iodine, you add the decolorizer for 5 to 7 seconds. Note that here, guys, I'm going to use my white. You're going to be like, I can't see that. Yeah, I know. So then you see that the specimen will be colorless. So you see how like you can see the line going right there? So yeah, this is going to be colorless, all right? Colorless because you allow the reagents to be poured out of the cell wall. So the cell is going to allow the reagents to be poured, the crystal violet and the iodine will be poured, released from the cell wall, from the peptidoglycan layer, from the porins, and then your cell is colorless. So if I were to go bring the slide to the stage and look at it through the microscope right now, I wouldn't see anything if it was a gram-negative microbe. All right. So that's why it's so important to make sure we finish this protocol. We're going to add our safranin. Sorry. So we're going to add our safranin. And with our safranin, we are going to see that your specimen should end up being that reddish pinkish color. All right. Reddish pinkish color. So overall, we can see the differences between gram-positive bacteria and gram-negative bacteria throughout the protocol for gram staining. All right. So let's see what that looks like. We have this rod-shaped bacterium, looks like a Tylenol capsule, and then you have this circle bacteria, right? So we add the crystal violet for 60 seconds, right? And then we're going to rinse with water. I want to cooperate. Let's see. Oh, there we go. 60 seconds for that one. Rinse with water, right? So we rinse with the water. And then after we rinse with the water, then we're going to apply the iodine. You cover that specimen with that. So notice how you cover the whole specimen, right? So you cover a good bit outside the specimen, on the specimen, of course, and around it too. So then after you rinse it with water, you add your iodine for 60 seconds. And then once you rinse your iodine off the slide, you're going to add your water. That's going to wash the slide. And then you're going to add your decolorizer, your alcohol. So once you add your decolorizer, your alcohol, that's going to run the color out of the rod-shaped bacteria. It's like, these are now colorless while the circles are purple. What does that tell us? What does that tell us about the bacteria? It tells me that, okay, the circles just might be gram positive and the colorless right now just might be my gram negative, but we won't know 100% until we add our last reagent, right? So we know that the alcohol is about 5 to 7 seconds. I say 5, lean toward 5. And then once you rinse it off, you're going to rinse it with water and then add your saffron in the counter stain for 60 seconds, right? And so what do we see? We end up seeing that the pink bacteria are in fact going to be the ones that were originally colorless, and these are representative of our gram negatives. And our purple bacteria are in fact gram positive bacteria. All right. So this is what it looks like if you have a mixed culture of a rod-shaped gram-negative bacteria. So if we have, for instance, E. coli, and then we have a coccus or spherical-shaped gram-positive bacteria. So like mentioned that with Staphylococcus epidermidis or Staphylococcus aureus, that's what this would look like, a mixed culture of Staph and E. coli. Pretty cool. All right, so here's some things to consider though when talking about the staining. So the gram stain is one of the most important staining techniques in medical microbiology, and it's important because it's used to diagnose different illnesses, right? It's very important for us to understand what is it so that we can treat it. That's really important. So the gram stain can be done to detect bacteria in clinical specimens, and it's also the first step, as I mentioned, for identifying an unknown. So you guys will talk about unknowns we will talk about unknowns in our class. And so with this gram stain, it can provide valuable information as well for treatment. All right, like I mentioned before. And so we're going to always have, or typically always have, our specimens as a fresh culture. And that's because gram stains are most consistent when used on young, actively growing bacteria. So freshly growing bacteria, so fresh cultures. All right. All right, so now we are going to talk about acid-fast staining. Okay, so acid-fast staining is another type of differential protocol. All right, so it's another type of differential protocol. And with this differential protocol, we're going to see that the bacteria, or should I say the stains, will bind only to bacteria that have a waxy material in their cell walls. And so this will not be decolorized by the acid alcohol, which is the decolorizing agent in this protocol. So I'll show you in a sec. That was a lot. I understand. We're going to break it down. Okay, but what I do want to take away for right now is that the acid-fast stain is used for identifying bacteria that are in the one of two genuses here, Mycobacterium and, no, well, genre. One of the two, you have Mycobacteria and Nocardia. So Mycobacterium should sound a little bit familiar because of what, TB, tuberculosis. So Mycobacterium tuberculosis is the causative agent of tuberculosis. All right, so when looking at the protocol, let's say we have a specimen. We don't really know much about it right now. We're trying to figure out what it is. So if we have this specimen, we're going to add the primary stain, which is carbofuchsin. Carbofuchsin is red when you look at it. And then we're going to add the decolorizing agent, acid alcohol, as well. And then we're going to add the methylene blue. And so with this acid-fast protocol, you have to understand that we're going to have to, I'm not going to make you guys know the timing on it, but I will tell you the timing on it. So of course you got to do your smear. And so for the carbofuchsin, typically when looking at this one, you have different protocols that can be used, different methods that can be used. And so when looking at this particular example, when you add your carbofuchsin to it, excuse me, it's only going to be on there for about like up to 5 minutes. All right, so only for about 5 minutes, up to 5 minutes. And so once you do that, you add your acid alcohol. Remember, that's going to be not that long. For this one, that's a little bit longer, a few seconds longer. And then for your counter stains, methylene blue, and that's going to be for a couple minutes as well. So with the acid fast bacteria, if it is in fact acid fast, it's going to stay red the whole time because remember, acid alcohol will not rush the carbofuchsin out of the cell membrane. So acid alcohol will not remove the carbofuchsin from acid-fast bacteria because they have the acid in their membrane. They have mycolic. Oh, I didn't tell you what it was. So the acid in their membrane is called mycolic acid. So in their membrane, they have this substance called mycolic acid, and that's why these guys are unaffected by the use of acid alcohol. Now, our non-acid-fast bacteria, if they go through this protocol, it's going to start off with the carbofuchsin being added to still, and it'll be red, but the acid alcohol will wash the decolorizer. Well, the decolorizer, aka the acid alcohol, will wash the carbofuchsin out of the cell because it doesn't have mycolic acid, right? And so this one will then retain the color of the last stain, the counter stain, methylene blue. All right, so it's going to retain that methylene blue pigment. So with the acid-fast stain protocol, it's typically used to diagnose TB, and that's basically when somebody comes to the hospital presenting symptoms, they'll do a really quick acid-fast stain as a confirmation based upon a certain presentation of symptoms. All right, so our signs and symptoms. And so figure 3-15, it shows us a light microscope of Mycobacterium tuberculosis. It's going to be staining that red pigment, as you guys can see right here. All right. Now we have special stains that are used to differentiate or distinguish parts of microorganisms. We're not going to do the protocol one, but we will look at those structures. So we'll look at capsule stains, endospore stains. We'll do the endospore stain though. So we actually do the endospore stain. We just don't do capsule stain or flagella stain. So for capsules, these are basically gelatinous coverings that's outside or external to the cell wall. So remember, we were talking about the cell, you got the cell wall structure, and then the capsule is going to be outside of that. Right, so that's going to be the capsule right here. And so with that capsule, it's going to not accept most dyes. And so that's why instead you have to do negative staining. So negative staining is when you stain the background in which your specimen is going to exist in. And so they use these acidic dyes, like we mentioned a moment ago, that's used for staining the background. So India ink and nigrosin, they can be used to contrast the background with the capsule structure. And so that's basically when you'll be simple staining these specimens. And so you'll look at the specimen and it'll look like it has like a little halo around it. So like the little white colors we see, like the halo that we see around the structures, that's the capsule. So stain the background. So that way you can see mainly the capsule structure of that particular specimen, these rod-shaped bacteria. Now, endospores are really interesting because these guys are really difficult to kill. So endospores are typically resistant dormant structures. So dormant means that they like stay inactive until activated by some type of external stimuli. And so when talking about these resistant structures, they're found inside of these cells and it's really difficult to stain them. They're not stained by ordinary methods. All right. And so when talking about the endospore stain, we're going to follow the Schaefer-Fulton method. The Schaefer-Fulton method is basically where you still have a primary stain, right? We're going to decolorize our cells and we're going to also have a counter stain. So I'll give you the time on those in lab. We won't do times right now because I really want you to focus on times for the gram stain. Gram stain times is my only concern right now. That's all I care about for lecture. So for the primary stain of the endospore staining protocol is malachite green. So malachite green, as the name tells us, is going to be green. And so you usually will heat the specimen too to help the dye penetrate the endospore. You're going to usually heat it, and that allows the dormant structure, right, that endospore, the coat to be breached, and the dye can actually reach the inside of it. All right, it's really hard to breach the coat, the endospore coat. So then you'll decolorize the cells with water. After you decolorize the cells with water, you're going to then apply the counterstain, safranin. And so safranin, as we mentioned before, is that reddish pinkish color. And so the spores will, the spores are going to look the green, have the green color. So the spores will have the green color, and then your cells themselves will be the reddish pink color. All right, so the cells will be the reddish pink. So let's look at that. So here we see the reddish pink color of our cells, and all those little circles, those little blobs of green, those will be your endospores. So those can be located within the cell, either on the terminal ends. So it'll either be on the ends, as you see right here, or it could be in the middle. It could be a central endospore. It just depends on what we're looking at. All right, and then the last one, the flagella stains. So flagella stains are basically allowing us to see the flagella. Now, the flagella is a structure that allows the cells to move, right? So structures of locomotion. Now, they're really slender, so it makes it kind of difficult to look at these guys just by using a light microscope. That's why we got to stain them. So the flagellar stain uses mordant and carbofusion to thicken the appearance of the flagella, and that's why we're able to see underneath the light microscope. And so why do we care about flagella staining? Well, that's going to be important when we start talking about enterics, gastrointestinal bacteria. Excuse me, because it allows us to differentiate between those guys, like how many bacteria, how many flagella do they have? How are they arranged? Are they only on the poles? Are they all around the body? Are they only are they only on one side? Or do they just not have them all together? Right. So this just shows us figures 3-16C, the flagellum structure present on these rod-shaped bacteria. All right, guys. So we have a few questions right here. I will read those questions for you guys, and you guys can pause it. And then if you want to wait for the answers, you can. All right, so which of the following is not a functionally analogous pair of stains? Is it A, nigrosin and malachite green, B, crystal violet and carbofusion, C, safranin and methylene blue, D, ethanol, acetone, and acid alcohol, or E, all of the above pairs are functionally analogous? All right, so pause it if you don't want the answer. The rest of us are going on. So the correct answer for this one is going to be nigrosin and malachite green. All right, nigrosin and malachite green for this particular one. All right. So which of the following pairs is mismatched? Is it A, capsule to negative stain, B, cell arrangement to simple stain, C, cell size to negative stain, D, gram stain to bacterial identification, or E, none of the above? All right, so you guys can pause it if you want to take some more time to think about it, but the rest of us are going to go ahead and go on in the interest of time. So the correct answer for this question is going to be B, as in boy, cell arrangement for the simple stain. All right. So thank you guys so much for tuning in. I truly appreciate your time, and I look forward to seeing you in lecture. As always, if you have any questions, please do not hesitate to reach out to me. Have a wonderful